Welcome to AP Biology. In this lecture, we are starting the cell cycle, how eukaryotic cells grow, copy their DNA, and divide through mitosis while keeping genetic information organized. This topic matters because every multicellular organism depends on accurate cell division for growth, repair, and reproduction. Next, we'll map out the big path we'll follow through the cell cycle. This roadmap shows the path we are about to follow. The cell cycle is not random. It is an ordered, regulated sequence that moves a cell through growth, DNA copying, and division. Interphase is the part of the cycle when the cell is active between divisions. In G1, the cell grows and does its normal jobs. In S phase, it replicates its DNA. And in G2, it makes final preparations for division. Some cells can also enter G0, a non-dividing state. That option matters because not every cell in your body is constantly moving toward division. After DNA replication, each chromosome has two identical copies called sister chromatids. That change sets up mitosis, when the duplicated chromosomes are separated accurately. Mitosis divides the nucleus, and cytokinesis divides the cytoplasm. Together, they produce two genetically identical daughter cells, which is the key outcome of this process. For AP Biology, do not just memorize the stage names. You will often need to interpret diagrams, match stage labels, and reason from cell count data about where cells spend their time. So keep two big questions in mind. What is happening to the chromosomes? And how does the cell make sure each new cell gets the right information? Next, we will sharpen the chromosome vocabulary you need for that. Before we talk about S phase and mitosis, let's lock in the chromosome vocabulary you'll need. The big idea is that DNA changes how tightly it is packaged, and that packaging affects what we can see during cell division. Chromatin is DNA packaged with proteins. When that chromatin coils up tightly for division, we call the condensed structure a chromosome. After DNA replication in S phase, one chromosome is not just one strand-like piece anymore. It consists of two identical sister chromatids, meaning two copied halves of the same chromosome. Those sister chromatids stay attached at a region called the centromere. They remain connected there until mitosis separates them into different daughter cells. In the image, focus on the idea of a diploid cell. It has two sets of chromosomes. Mitosis preserves that set number, so the daughter cells keep the same chromosome number as the original somatic cell. This is a key AP Biology distinction. Mitosis maintains chromosome number in body cells, also called somatic cells. Meiosis is different because it reduces chromosome number to make gametes like sperm or eggs. Concept check. After DNA replication, which description correctly identifies one chromosome? Choose the option that describes one replicated chromosome, not a homologous pair and not separated chromatids. A is correct. After DNA replication, one chromosome consists of two identical sister chromatids connected at a centromere. B is tempting but wrong. Homologous chromosomes are a pair from two parents, not identical sister chromatids of one replicated chromosome. Keep that picture in mind. Replicated chromosomes have sister chromatids joined at a centromere, and mitosis separates those chromatids while maintaining chromosome number. Next, we'll place that event into the larger ordered cycle of the cell. The main idea here is that cell division is not a random scramble of events. A cell moves through an ordered cell cycle, meaning each major event happens in a regulated sequence. Start with the sequence on the left. The cell grows, prepares for division, copies its DNA, and then separates those chromosome copies. That order matters, because separating chromosomes before copying DNA would not produce healthy daughter cells. Now look at the circular model. The circle helps you see that the cell cycle repeats. After one round finishes, the new cells can enter the cycle again if conditions allow. The cycle is organized into two broad parts. Interphase is the long preparation part, and it includes G1, S, and G2. The mitotic phase is the division part, and it includes mitosis plus cytokinesis. In interphase, the cell is active. It grows, copies its genetic information during S phase, and gets ready to move chromosomes. In the mitotic phase, mitosis separates the chromosomes, and cytokinesis divides the cell's cytoplasm. The usual outcome of one complete cell cycle is two daughter cells. Daughter cells are the cells produced by division, and each can begin the cycle again when the right signals are present. So keep this big structure in mind. Interphase prepares, and the mitotic phase divides. Next, we will zoom in on the three parts of interphase. Interphase is the part of the cell cycle where the cell does the behind-the-scenes work for division. It is not a resting stage. It is active preparation. In G1, the cell is metabolically active, meaning its chemical reactions are running at a high level. The cell grows, makes more cytosol, and duplicates organelles so both future daughter cells can receive enough cellular parts. During S phase, the S stands for synthesis, because the cell synthesizes, or copies, its DNA. The DNA is still loosely packed as chromatin, which makes it accessible for replication. After replication, each chromosome has two identical sister chromatids. These sister chromatids stay attached at a region called the centromere until they are separated later in mitosis. In G2, the cell checks and finishes its preparations. It produces proteins, builds up ATP reserves for energy, and gets ready for the physical movement of chromosomes. A key part of that preparation is duplicating centrosomes. Centrosomes help organize the mitotic spindle, the structure that will pull sister chromatids apart during cell division. So, think of interphase as a three-part preparation sequence. Grow in G1, copy DNA in S phase, and stock up and organize in G2. Next, we will see what it means when cells step out of this cycle instead of continuing toward division. Cells do not always keep moving toward division. G0 is a non-dividing state, meaning the cell is outside active preparation for DNA replication and mitosis. A cell in G0 is not dead or inactive. It can still do its normal job, use energy, make proteins, and respond to its environment, but it is not cycling toward division. G0 can be temporary. For example, liver cells and some immune cells may pause, then re-enter the cell cycle when the body needs repair, growth, or a stronger immune response. One kind of signal that can call some cells back into the cycle is a growth factor. A growth factor is a signaling molecule that tells a cell conditions are right to grow and divide. Other cells enter G0 for the long term. Many neurons and cardiac muscle cells are highly specialized, so they often stay non-dividing instead of repeatedly replacing themselves. This is why different tissues have very different division rates. Skin and intestinal lining cells divide often, but many brain and heart cells divide rarely, partly because of long-term G0. So remember, G0 is not simply resting. It is a regulated exit or pause from the cycle, and next you will use that idea to check your understanding of interphase and G0. Let's pause and check your understanding of interphase and G0. Focus on the order of the cycle, what happens in each interphase stage, and how G0 differs from simply resting. Which sequence correctly shows the main order of events in the cell cycle, starting with G1? The correct order is G_1, S, G_2, mitosis, and cytokinesis. During interphase, the cell grows in G_1, copies DNA in S, and prepares for division in G_2. Mitosis and cytokinesis follow. A tempting mistake is placing G_2 before S, but the cell must replicate its DNA before final preparation for division. Which option correctly matches G1, S phase, and G2 with their major events during interphase? In G_1, the cell grows and carries out normal functions. In S phase, DNA is replicated, and in G_2, the cell makes final preparations for mitosis. The most tempting distractor is the idea that G_1 is when DNA replication occurs, but DNA copying specifically happens during S phase. Which statement best distinguishes interphase from a true resting or non-dividing state? Interphase is not a true resting state. It is an active period that includes growth, DNA replication, and preparation for cell division. The tempting misconception is that interphase means the cell is inactive, but cells in interphase are metabolically active and progressing through the cycle unless they exit to G_0. Which statement best describes G0 in multicellular organisms? G_0 is a biologically important pause or exit from the cell cycle. Some cells may re-enter the cycle when they receive the right signals, while others may remain in G_0 long-term. A common mistake is thinking G_0 means the cell is dead, but many G_0 cells are alive and specialized. Start the cell in G1, the growth phase before DNA replication. At this point, the cell has one complete genome, so its relative DNA amount is 1 times, and the chromosome is still unreplicated, not split into sister chromatids yet. Now move into S phase. S stands for synthesis, meaning DNA is being copied, As replication happens, the DNA amount rises from 1 times toward 2 times, and each chromosome becomes a duplicated chromosome made of 2 sister chromatids joined together. By G2 and the start of M phase, replication is complete, so the cell has 2 times the DNA for a short time. During mitosis and cytokinesis, the sister chromatids separate and the cell divides, leaving each daughter cell with 1 times DNA, which means 1 complete genome. Keep that pattern in mind as we zoom into mitosis next. Mitosis is the part of the cell cycle that separates duplicated chromosomes into 2 identical daughter nuclei. The goal is simple but essential. Each future cell needs a complete genome, meaning a full set of genetic information. Start at prophase. The duplicated chromosomes condense into visible sister chromatids, which are identical DNA copies attached together, and the spindle begins forming to move them. Next is metaphase. The chromosomes align at the cell's equator, called the metaphase plate, so each duplicated chromosome is positioned for an even split. In anaphase, the sister chromatids separate. Once they pull apart, each chromatid becomes a chromosome, and complete sets move toward opposite poles of the cell. In telophase, new nuclear envelopes form around the separated chromosome sets. The chromosomes begin to decondense, and the cell now has 2 daughter nuclei with matching genetic information. These stage names make mitosis easier to describe, but the process is continuous, not a set of sudden jumps. What really matters is accurate spindle attachment and clean chromatid separation, because that is what preserves 1 complete genome for each daughter cell. The small final arrow points to cytokinesis, when the cytoplasm divides. We'll separate that idea from mitosis next, because mitosis divides the nucleus, while cytokinesis divides the cell body. After the chromosomes have been separated into 2 nuclei, the cell still has one more big job. It has to split the cytoplasm so 2 separate daughter cells can form. Cytokinesis usually follows telophase. The word cytokinesis means division of the cytoplasm, so this is the physical separation of the cell body, not the sorting of chromosomes. Keep this distinction clear. Mitosis divides the nucleus, while cytokinesis divides the cell body. They are connected parts of cell division, but they are not the same process. In animal cells, cytokinesis happens by pinching inward. A contractile ring, made of protein fibers, tightens under the cell membrane and creates a cleavage furrow, which is the groove that squeezes the cell into 2. Plant cells cannot simply pinch inward because they have a rigid cell wall. Instead, they build a cell plate down the middle, and that cell plate develops into a new cell wall between the 2 daughter cells. Use the comparison table as a quick memory tool. Animal cells form a cleavage furrow, and plant cells form a cell plate. Different mechanism, same goal, separate 1 cell body into 2 cells. Now check your understanding. Which statement correctly distinguishes mitosis from cytokinesis? Choose the option that separates nuclear division from cytoplasmic division. B is correct. Mitosis separates duplicated chromosomes into daughter nuclei, while cytokinesis separates the cytoplasm and cell body. C reverses the mechanisms. Animal cells use a cleavage furrow, and plant cells form a cell plate. As you move forward, keep asking what is being divided, the nucleus during mitosis or the cytoplasm during cytokinesis. That distinction will help explain why daughter cells end up complete. Mitosis preserves chromosome number because division is carefully prepared before it happens. First, the cell makes a matching copy of every chromosome, and only then does it separate those copies. During S phase, the DNA synthesis phase of interphase, each chromosome is copied. The 2 copies stay attached as sister chromatids, meaning identical DNA copies joined together as 1 duplicated chromosome. Now mitosis has something organized to work with. Each duplicated chromosome has 2 matching chromatids. That matching pair is the key setup for equal distribution. During mitosis, spindle fibers, which are protein structures that move chromosomes, line up the duplicated chromosomes and then pull sister chromatids toward opposite poles of the cell. Because each sister chromatid goes to a different side, each forming daughter nucleus receives 1 chromatid from every duplicated chromosome. In other words, neither nucleus should be missing a chromosome copy. So the big AP biology claim is this: because sister chromatids are copies, the daughter cells receive complete, matching genomes. A genome is the full set of genetic information in the cell. That outcome depends on accuracy. If no mutation changes the DNA and no segregation error sends chromatids to the wrong place, mitosis produces genetically identical daughter cells, which is why it is so useful in the body. Mitosis is not just a neat chromosome sorting process. Its bigger job is to let organisms build, maintain, and sometimes reproduce by making new cells with the same genome. First, mitosis supports growth by increasing cell number. An embryo starts as one cell, and repeated rounds of mitosis help build the tissues and organs of a developing body. Mitosis also maintains tissues. Your skin constantly replaces worn or damaged cells, and the intestinal lining turns over rapidly because those cells face a lot of friction and chemical stress. In many eukaryotes, mitosis can even produce whole organisms through asexual reproduction. Asexual means one parent is involved, and the offspring inherit that parent's genome, so they are often genetically very similar. The timing of the cell cycle varies a lot. Some mammalian cells grown in culture divide in about 24 hours, but some specialized cells enter G0, a non-dividing state, and may stay there for a lifetime. So when you see mitosis, think beyond the phases. It is how cells pass on chromosomes while supporting growth, repair, replacement, and asexual reproduction. Next, you'll get a chance to check these ideas with a few questions. Let's pause to check your understanding of mitosis, chromosome movement, and why this process matters. Choose the best answer for each question before reading the feedback. Which sequence gives the correct order of the 4 main stages of mitosis? The correct order is prophase, metaphase, anaphase, telophase. A common mix-up is placing anaphase before metaphase, but chromosomes must first align at the cell equator in metaphase before sister chromatids separate in anaphase. During which stage of mitosis do chromosomes line up at the equator of the cell? Chromosomes align at the cell equator during metaphase. Prophase is tempting because chromosomes condense and the spindle begins forming, but alignment at the middle happens after that, in metaphase. Why does separating sister chromatids during mitosis help each daughter cell receive a complete genome? After DNA replication, each sister chromatid is one complete copy of a chromosome. When sister chromatids separate, each daughter nucleus receives one copy of each chromosome. Choice B is a tempting misconception, but daughter cells do not receive chromosome halves and rebuild them during telophase. Which statement correctly distinguishes mitosis from cytokinesis? Mitosis is nuclear division. Replicated chromosomes are separated into daughter nuclei. Cytokinesis divides the cytoplasm, producing separate daughter cells. Choice A reverses the roles of the two processes. Which example best shows mitosis contributing to growth or repair in a multicellular organism? Replacing skin cells after an injury is a clear example of mitosis supporting tissue repair. Choice D is related to the cell cycle because DNA replication occurs before mitosis, but S phase itself is not the process that separates chromosomes to make new nuclei. Use this as your quick reference sheet for the cell cycle. The main idea is to connect each phase with what the cell is doing, and then connect the chromosome vocabulary to what moves during mitosis. In G1, the cell grows and makes the molecules it needs, a process called biosynthesis. In S phase, the cell copies its DNA, and in G2, it makes the final preparations for division. Mitosis is nuclear division, meaning the copied chromosomes are separated into 2 nuclei. Cytokinesis is cytoplasmic division, where the rest of the cell splits, and G0 is a non-dividing state that some cells enter. Now match those phases to the chromosome terms. Chromatin is the loose DNA and protein complex, while a chromosome is the condensed DNA package you can track during cell division. Sister chromatids are the identical copied DNA molecules formed after replication. The centromere is the attachment region that holds sister chromatids together until they separate. Spindle fibers are the structures that move chromosomes during mitosis. The centrosome helps organize those spindle fibers, so chromosome movement is accurate. The outcome to remember is that mitosis and cytokinesis produce daughter cells. If chromosome separation is accurate, those daughter cells are genetically identical and each has a complete genome, meaning a full set of genetic information. As you study, try covering one side of each definition and testing yourself. Next, we will pull these terms together into the final takeaways. To wrap up, connect the vocabulary you just reviewed into one big idea. The cell cycle is how eukaryotic cells grow, copy their DNA, and pass chromosomes forward accurately. The cell cycle is ordered and regulated, meaning its steps happen in a controlled sequence. That control supports growth and reproduction while keeping chromosome information continuous from one cell generation to the next. Interphase is the preparation part of the cycle. In G1, the cell grows and builds needed materials. In S phase, it replicates its DNA. And in G2, it finishes getting ready for division. G0 is different because the cell is not actively dividing. Depending on the cell type and the signals it receives, G0 can be temporary, like a pause, or long-term, like a specialized working state. Mitosis is nuclear division, and its job is to separate sister chromatids. Prophase condenses chromosomes and builds the spindle. Metaphase lines chromosomes up. Anaphase pulls sister chromatids apart, and telophase reforms nuclea. Cytokinesis finishes the process by dividing the cytoplasm. The result is 2 daughter cells, each with a complete genome, meaning a full set of genetic instructions. So the big takeaway is this: accurate cell division depends on preparation, chromosome separation, and final cell splitting all working together. If you can explain that flow, you understand the core purpose of the cell cycle. Nice work finishing this lecture.